Hello, my name is Vedant Kansara, and I'm from Farmington High School in Farmington, Connecticut. Today, I will present my research on age-related myeloid cell states in glioblastoma. Glioblastoma, or GBM, is the most common and most aggressive malignant brain cancer in adults, with an average survival time of only 15 months. Despite the standard of care treatment consisting of surgical tumor removal followed by chemotherapy and radiation, GBM universally recurs. In older patients, GBM is even more aggressive, with an average survival time dropping below 9 months. GBM grows so quickly due to its high infiltrative ability and difference in cellular makeup between patients. However, GBM also grows rapidly due to its microenvironment, the cancerous and healthy cells surrounding the tumor in the brain. Within this microenvironment are myeloid cells, a family of immune cells including microglia that provide immune surveillance and resolve tissue damage during diseases. All microglia support homeostasis and protect the brain, but each microglial cell expresses a unique combination of genes based on a specific role, known as its cell state. Past studies of microglia in the healthy or non-GBM environment have suggested that aging-related immune decline may be a possible cause of decreased survival with age. However, age-related changes in patients with GBM remain understudied. I utilized microglial cell states from two past studies on microglia in the healthy brain, Hammond et al. And Safian et al. Hammond et al. Found four age-related microglial cell states, aging 1A, 1B, 2, and 3. They found increased inflammatory markers in aging 2 and increased interferon or disease and infection related markers in aging 3, suggesting that those two cell states may be related to causing neuroinflammation and degeneration of brain tissue in older patients. Safian et al. Studied microglia in the white matter, which is primarily deep within the brain. They also found four distinct cell states, two of which were age-related. They found white matter-associated microglia, known as the WAM cell state, an activated cell state, and two homeostatic cell states. The WAM and activated cell states increased in the microglia with age, and they both upregulated disease response, lipid metabolism, and phagocytosis genes. My objective was to leverage these 8 known microglial cell states from healthy aging and investigate whether or not they were conserved during microglial aging in older patients with GBM. So, I followed 3 basic steps in my research methodology: data processing, cell clustering, and finally, mapping aging-related cell states. For data processing, I used data from two mice GBM samples, one young mouse, which was 3 months old, and one old mouse, which was 24 months old, and both were injected with tumor cells, and single-cell sequencing on these two mice was performed. This resulted in a cell-by-gene matrix of over 1.6 million brain cells. I pre-processed the data and filtered out cells with more than 10% mitochondrial transcripts, as well as less than 250 genes, which left 21,376 usable high-quality cells. Next, I used unsupervised clustering to find and visualize specific cell populations through UMAP, which is shown on the left. Each dot represents a cell, and each colored group represents a cell cluster. To identify the populations, I then used the Azimuth platform from the Human Biomolecular Atlas program to reference map the dataset with known mouse gene expression data. I also ran the infer-CNV package, as shown on the right, which generated a plot of chromosomal deletions in blue and chromosomal additions in red to increase confidence in cell and tumor clusters. Using the high-quality cells I found many cell populations, including neuronal clusters, astrocytes, oligodendrocytes, immune populations, and my target microglia population. I also identified tumor cells due to an increased expression of GBM-related genes, such as VIM, SOX2, OLIG1, and HEX-B. I then subsetted the target microglia population from the data, which left 984 microglia from both the tumor-bearing and non-tumor hemispheres of the aging and young mouse brains. Finally, using the microglial cell states from Hammond et al. And Safine et al., I gave each microglial cell in my data a score from 0 to 1 for how similar its gene expression was to each of the 8 known cell states. This means each microglia would have 2 states, one from Hammond et al. And one from Safine et al. Based on this, I measured the proportion of microglia in each state in the young and old samples and visualized the most differentially expressed genes between the two populations. From Hammond et al., I combined aging states 1A and 1B into one cell state as they had transcriptionally similar signatures, meaning they upregulated and downregulated the similar genes. Plotting these cell state proportions in the microglia showed that the aging 2 signature increased with age, as shown in the black shaded section, from 0.97% in young microglia to 27.59% in old microglia. The cell state aging 3 also increased with age, from 2.32% in young microglia to 23.89% in old microglia, as shown by the increase in the gray shaded section. In contrast, the aging 1A/1B cell state significantly decreased with age, from 96.71% to 48.48% in old microglia. Because cell state aging 2 had upregulated expression of inflammatory markers, these results suggest an increase in the inflammation in the older brain with GBM. This increase indicates that older microglia release more inflammatory and cytotoxic signals, which could damage healthy brain cells and promote tumor proliferation in the older brain. Similarly, because the aging 3 cell state upregulated expression of many interferon or disease-related response genes, my results suggest that there's an increased disease/infection response in old microglia, further corroborating heightened immune activity. This could cause excess inflammation and cytotoxicity, harming the aging brain and accelerating GBM growth. I repeated this process with the cell states from Safine et al., and I combined cell states homeostatic 1 and 2 because they had transcriptionally similar signatures. Plotting these cell states revealed the surprising finding. The activated microglial cell state signature decreased in proportion with age, as shown by the decrease in the black shaded section, from 33.14% in young microglia to 0.00% in old microglia. Additionally, the WAM signature decreased in proportion with age as well, from 15.31% in young microglia to 4.26% in old microglia. The WAM and activated cell states have been shown to upregulate expression of lipid metabolism, disease response, and phagocytosis genes. WAM microglia have also been shown to have an increased ability to metabolize and digest myelin. However, my results suggest that in contrast to healthy aging, where WAM and activated states increase with age to compensate for increased myelin buildup, there is a lack of WAM microglia in the diseased brain. This leads to excess myelin debris and eventually buildup, which could have many detrimental effects, such as neurodegeneration, brain impairment, and brain damage, all of which can allow GBM to grow and recur faster in older patients. I performed further differential gene expression analysis to find enriched genes in the aged microglia, and this revealed 103 significantly enriched genes, many of which overlapped with those from Hammond et al. And Safine et al. There was a high overlap of inflammatory and interferon response genes, such as H2AB1, H2AA, CD74, and GBP2, which overlapped with cell states 2 and 3 from Hammond et al. And the WAM state from Safine et al., further supporting increased immune activity in the older brain. In this project, I identified age-related shifts in microglial cell states in the GBM environment towards increased immune activation and interferon signaling that could be causing neuroinflammation and brain damage. These factors can cause excess cytotoxicity within the brain, damaging healthy cells and allowing GBM to grow and recur in older patients. Concurrently, I found that microglia shift away from homeostatic maintenance in the older brain with GBM, which can cause a buildup of myelin debris and exacerbate neurodegeneration, further promoting tumor proliferation and infiltration in the aging brain. These results suggest two potential paths for future studies. One, limiting microglial activation and pro-inflammatory pathways may be a potential target for GBM control, as it can limit the immune response and prevent excess damage to healthy brain cells. Two, increasing formation of white matter-associated microglia may also be a potential treatment for GBM in older patients, as WAM microglia could prevent the buildup of debris and myelin, which are primary causes of neurodegeneration. In the future, I want to extrapolate these findings to human samples and see if there are age-related changes in human microglia. I also want to investigate cell state changes in other immune populations to find new methods for combating tumors in older patients. I would like to take this time to thank my mentors, Frederick Varn and Megan Callender, for their help and invaluable guidance in this project. I would also like to thank JSHS for this fantastic opportunity. Thank you.